In this video, we will discuss about FAS disruption, and by targeting ITAM signaling pathways can enhance the NK CAR anti-tumor response to a great extent. So, how these researchers come up to these ITAM signaling pathways? As previously, they published a study where they have seen that in the T cells, by targeting these ITAM signaling pathways, it will enhance its anti-tumor efficiencies. So they are expecting that this ITAM will also enhance the NK CAR cells anti-tumor efficiencies if this ITAM is been targeted through this CAR. So, and we have seen in our previous videos that this FAS disruptions increases the survival rate of the CAR cells. So what they test in this case is that they first, in the panel A, it shows their map where they do not disturb this FAS signaling pathways. They just introduce this ITAM in the CAR and check its efficiencies, and they check it with the 3x time ITAM and 1x time ITAM in the intercellular domains, and in the high ET ratios they do not see any significant difference, but in the low ET ratios, that is low effector to target ratios, it means the effector cells are low and target cells are high, they are able to see significant changes that can be seen here. And so because of this results, they thought that ITAM is actually also working in the NK cells at low effector to target ratios. Thereafter, they wanted to check its efficiencies in the in vivo studies. So they took the mice and inject this mice with null M6 tumor cells, and one day after that they transfused these NK cells at high effector to target ratios and then check its observations. And what they're able to see is that at high effector to target ratios, the difference between the control, that is in which they use the PBS, and in the case where they only use the CAR construct, that is TEGFR, they do not see any significant changes in the survival rate. But when they used CAR construct with this ITAM, 1x ITAM, or this not 1x, this ITAM signaling pathways, that is 1XSS TEGFR and the 1XSS TEGFR plus delta FAS has been disturbed. In both the cases, they are able to see a high survival rate, significantly high survival rate in the high ET. And when they do the similar studies at low ET, that is they in this case they transduce these NK cells around 10 raised to 10 raised to power 6, but they transduce them 4 days after the mice is been induced with this null M6 cancer. In the first case, it is just 1 day after that, and in the null M6 case, the tumor cells that are injected is also higher, it is 10 raised to power 5, and in the previous case, the 2 into 10 raised to power 4 null M6 tumor cells are injected. So this makes it a low effector to target ratios, and in that case, they can clearly see the difference between ITAM signaling CAR with ITAM signalings and no change in the FAS, whereas with CAR with ITAM signaling, targeting ITAM signaling pathways with FAS has been disrupted. So the case where FAS has been disrupted shows a significantly higher survival rate compared to the case where ITAM CAR is transduced but no changes in the FAS. So from that, they come to a conclusions, and their final theory based on their observations is shown in this panel D, where if the CAR has this FAS-L gene present, then once it gets activated by these tumor cells, what will happen is that the FAS-L receptors will also interact with the FAS-L receptors also get interacted, and that will results in the CAR cell apoptosis. And in that case, if this FAS-L's signaling has been disturbed or somehow been changed such that FAS intracellular signaling does not get triggered, then it will prevent the apoptosis of the CAR T cells, and it will cause the tumor cells response more. It will have a more efficient response against the tumor cells. In the case where FASLG has been knocked down, they cannot interact with the intracellular FAS signaling pathways cannot be triggered, and in that case the CAR cell survival rate is higher, and it can act on the tumor cells with its highest efficiencies, or much higher efficiencies compared to the case where FASL gene is present in wild type. And similarly, if somehow its receptors also get disturbed, FAS receptors on the CAR T cells also get disturbed, that will prevent the intracellular signaling pathways. And in that case also the effect of these CAR T cells against the tumors will be higher compared to the case where FAS's intracellular signaling has been triggered. So in the bottom two cases we do not see any significant changes, but in the top two cases we will see significant changes, as the cell apoptosis will be reduced in the because of the intracellular signal triggering of this FAS-L signaling pathways. This is what they finally conclude. So I hope you understood the story and the results that they have researchers have presented in the main figure. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.